Record to the cloud. Okay, so we are going to begin the lymphatic tissue. Okay, so this is what we're starting today. So first of all, about the lymphatic tissue. Lymphatic tissue is pretty much synonymous with the word immune system, okay, with the words immune system. So both of them are related to one another. So whenever you're actually saying lymphatic tissue, you're kind of thinking about immune system as well, because what is the major cell of the lymphatic tissue? It is going to be a lymphocyte, and as you know, lymphocytes, they are immune cells, right? So they actually belong to immune cell. But last semester in histology, we kind of touched lymphatic vessel, right? So specifically lymphatic vessel. Maybe you remember, what is the job of lymphatic vessels? What do they do? Huh? Lymphatic vessels. For sure you guys know, so come on.
They carry the tissue fluid?
Very good. So it's better, they do carry, but the first thing you need to say is that they collect the excessive fluid, right? So the excessive fluid of the connective tissue will be collected by the lymphatic vessels. You have to remember that they begin as the blind sacs, okay? So lymphatic vessels, they simply begin as this blind-ended sac of some sort, which is just actively collecting whatever excessive amount of fluid you have within the connective tissue. And then they carry that lymphatic fluid to where? Where do you eventually drop all of this lymphatic fluid? You drop it to your veins, okay? So eventually all of the lymph, whatever lymphatic fluid you have, eventually all of it will be it will end up within your veins, but we'll get more details there as we go. Okay, so we have said that the main cell in the lymphatic tissue is going to be obviously a lymphocyte. Everybody knows the function of lymphocytes, right? So they belong to the immune system. So they're just protecting you from potentially harmful substance. Where can we find those lymphocytes, right? So where can they be? So they can be in the solid organs, for example, like your lymph node, right? Spleen, bone marrow, thymus, or they can be simply diffuse. They can be everywhere. So we say diffuse lymphatic tissue. When I say diffuse lymphatic tissue, what am I actually talking about? Maybe you can kind of guess. Diffuse lymphatic tissue, the lymphocytes which are everywhere. How do we call it? In our first class, we discussed this. First class ever with us.
So lamina propria.
Perfect, lamina propria. So diffuse lymphatic tissue, another way to describe it is simply to say lamina propria. Even another way to say it is MALT, mucosa associated lymphatic tissue. All three things are the same. They're talking about the same thing. So diffuse lymphatic tissue is lamina propria. It is also mucosa associated lymphatic tissue, or simply MALT. Okay, so I hope that is clear. So they're diffuse, they're not solid organ-like substances, they're just diffuse, they're just everywhere, okay? Now, we have two types of the organs, the lymphoid organs. We have the primary and we have the secondary. Another name for primary are central, another name for secondary, peripheral. So what is the difference? So first of all, which lymphoid organs are primary? This is your bone marrow and thymus only. So only the two areas are considered to be central or primary. Okay, so bone marrow and thymus. And what is happening in them? You can think about those organs as a school for lymphocytes. This is the area where the lymphocytes are being educated. This is their school, this is their university. All other organs, the ones that we saw here, so your lymph nodes, spleen, your lamina propria, right, your tonsils, all of them are going to be secondary lymphoid organs. And what can you think about them? This is the place where the lymphocytes go to work. This is their job place. So primary lymphoid organs is the place where your lymphocytes get educated. This is where they learn This is where they learned how to be a lymphocyte, what to do, and etc. Secondary lymphoid organ is time to apply your knowledge. Whatever you have learned in the bone marrow and thymus, time right now is to actually apply your knowledge, right? To, I don't know, detect an antigen, kill it, and etc. So I hope that is clear. We have two types of immune responses, right? So, for example, I don't know, your immune system has encountered a certain bacteria or a virus or a pathogen of some sort, right? So there are two ways to respond to that. One of them is called innate, and another is called adaptive. Innate is also known as the non-specific, adaptive is known as the specific. What is the difference? Innate immune response is very primitive. It's extremely primitive. It's something that you were already born with. Okay, so when you were born as a baby, you already had your innate immune response. The adaptive is in the name. It's something that you adapt to. For example, typically, not always, but typically, once as a baby, I got my chicken pox, right? I have been sick with a chicken pox. In my adult life, I typically do not get sick again. Again, I say mostly because there's some cases where you get reinfected with chicken pox. It's very rare. So what am I trying to tell you? In the adaptive immune response, your immune system remembers the moment it has encountered something pathogenic. So if for innate immune response, every time is like a new time, it doesn't matter how many times they encounter that bacteria or virus, every time it meets it, it's the same as it's new. I have never seen this bacteria ever before in my life, right? Even though it encounters that bacteria for the 1000th time already. The adaptive has a very important keyword, which is a memory. Okay, so adaptive immune response possesses a memory. So once is enough. So whatever belongs to the adaptive immune response, right? So once you have it, once you have encountered bacteria through adaptive immune response, you're going to see that you will remember it for the rest of your life. So this is why, which one are you born with? You're born with the innate. This is very primitive. The one that you acquire as you grow up is the adaptive. Which cells belong to the immune system? Well, all of them are here, but we'll talk more details about them as we go. Today in our lecture, we're going to have 4 crucial slides. Okay, so mark it for you that you have to know it for oral, you have to know it for self-control test. It's the most crucial term that there is. And the first crucial slide is this one, the one that says antigen. You have to understand what is it, because in your oral, they will typically ask you about it. So it doesn't matter which slide you pick. In that slide, there's always something immune, some kind of cell which is immune. So they will typically drag you to this area right here. So antigen, maybe somebody already knows. What is antigen? Come on, guys, don't be shy.
It's like a label for like production of antibodies, basically.
Perfect. So antibody is always against antigen. So whatever antibody you're making, it's always against antigen, always. So that being said, that antibody is against antigen. So what is antigen? Antigen is a marker of some sort. So if you have to describe what is antigen, it is a marker. Now, I'm going to give you 2 definitions for antigen. One of them is this year for anatomy department. Okay, so this is what they want you to say. One of them is next year for immunology. So when you're going to be 3rd year, you're going to have immunology as a subject, and you will also talk about antigen there. The reason I need to give you 2 definitions, because the one that anatomy tells you is wrong. So when you say it into the immunology professor, they laugh in your face and they say, who has taught you this? Because this is absolutely wrong. So I'm not sure if you want to hear the 3rd one in order not to confuse, because I have experienced that some students don't want to hear about the 3rd year definition of antigen? Do you want to or you don't want to? Do you want me to just tell you how anatomy department says it, or do you want the actual one?
We want that.
Fine.
Okay, I did not understand anything.
Both.
So,
Both. Just give us
both?
both.
Okay, let's do both. So first of all, for anatomy department. For anatomy department, antigen is everything foreign, everything that is not yours. So it's a foreign substance. It's a foreign marker of some sort. And this foreign marker can definitely induce immune response. This is very important, okay? So antigen will always induce immune response, always. And what can induce immune response? Well, definitely something foreign, something that is not your body's, right? So for example, if there's a bacteria of some sort, on its surface, there's going to be a marker, and that marker says, I don't know, E. coli bacteria, right? So that's antigen. So it will be a protein or a sugar or I don't know, a toxin of some sort, but that's something which is on the surface of bacteria or virus or parasite is called antigen, right? So again, antigen is like a receptor or like a marker that is typically always foreign. And because it is foreign, it can induce immune response. So this is for anatomy department. But you have to understand, my own cells in my body, they also have a marker. For example, if I take a look at my epithelial cells, each of my epithelial cell has a marker that says it is an epithelial cell. I look at my cardiac myocyte, each and one of them has a marker that says it's a cardiac myocyte, brain cell, I don't know, red blood cells, it doesn't matter, right? So each cell in my own body also has a marker that tells me what is it. So in immunology, you call that self-antigen. So do you see where is the difference? For anatomy department, antigen is always foreign. It can never be in your own body. But for immunology, there is. In your own body, your own cells, they also have markers that tell you what they are. You just don't call them antigens, you call them self-antigens. Does it make sense? Yes? No?
Yes.
Yes, yes.
Yes. Okay, so imagine the following situation. I am Yulia. In my own body, I have lymphocytes. What would you find on my own lymphocytes? A marker that says I am Yulia's lymphocyte, right? It's my lymphocyte. That marker, is it self-antigen or simply antigen?
Self-antigen.
Self-antigen because it's mine. Those are my lymphocytes. And then, I don't know, I take a sip of this water, and this water contains influenza bacteria. On the surface of that influenza bacteria, I will also have a marker that tells me this is bacteria. How do you call that marker?
Antigen.
Antigen. Which one will induce immune response? Antigen. How do you call disease when self-antigen induces disease, induces immune response? How do you call that?
Autoimmune.
Perfect. You call it autoimmune disease. So autoimmune disease is when my own lymphocytes see, like, I don't know, my kidney, and they're like, oh my God, this kidney definitely looks like a virus. I must kill it. Okay, so this is the autoimmune disease. So I hope that was clear. So antigen, because we're second year medical students, is foreign. Okay, so we're not third year, we're second, so antigen is foreign, right? So it's a foreign substance, is a foreign marker that is present on everything that is not your body. I'm talking about on the surface of a peanut. Each peanut consists of a bunch of cells. Each of a cell that makes up a peanut has a marker that says it is a peanut. How do you call that marker? You call it antigen. What can be an antigen? Foreign protein, polysaccharide, toxin, an entire infectious organism, foreign tissue, or transformed tissue. Transformed tissue is cancer, Okay, so even your own, for example, cancer cells that are within your own body, they could still be considered as foreign because that's not supposed to be there, right? So speaking of the two immune responses, so we're back to it. We have the innate immune response and we have the adaptive immune response. So we have said innate is primitive. It's something that you're born with. So what are its parts? Physical barrier. I mean, skin is there for a reason, to protect me from whatever, I don't know, potential pathogens there are, right? Mucous membrane, so your mucosa, which is lining your gastrointestinal tract or your urinary tract or, I don't know, your respiratory tract, it's also going to be protecting your body from within. So if skin protects you from outside, mucosa protects you from within. It makes sure that nothing can easily diffuse. Chemical defenses, so low pH, acidic pH. So for example, in the stomach, what is the pH? Somebody knows? What's the pH of the stomach?
Around 6.
Ooh,
2.
two.
Around
Actually
2.
two. It's actually 2. It's super acidic, okay? It's super acidic. So the pH is 2. It's very, very low, so it's quite acidic. In your esophagus, for example, the part which is closer to the stomach, there it is 6.5, okay? So it's almost normal, like balanced out. In vagina, for example, it's also a very acidic environment there. The pH is about 4.5 to 5, okay? So it's also acidic. Now, various secretory substances, none of them you need to know. They were all in your book. More details on that in your third year immunology. The one that you do need to know is immunoglobulin. Which immunoglobulin is in your secretions?
IgA.
IgA, okay? So don't forget that IgA is going to be within your secretory substances. It is in your tears, in your sweat, in your saliva, right? So in all of those things, and it will definitely protect you from whatever is coming in. And now the question that will be asked in the self-control test is which cells belong to the innate immunity response. So macrophages, they come from monocytes, right? So monocytes are also going to be part of it. All granulocytes, so not just neutrophils, also basophil, also eosinophil. And this one is the one that they want, natural killer cell. We will talk about it today, but you have to understand that natural killer has more in common with B and T lymphocytes rather than with those cells here. So this is why they typically test you on this in order for you to understand this, right? So natural killer, even though it's more related to lymphocytes, it is not part of the adaptive immunity, it is part of the innate immunity. You're already born with a bunch of natural killer cells. Again, we'll talk more details about it today. Adaptive immunity, do not forget that it has a very specific response. The key word for it is a memory, right? So it remembers the encounter, it remembers what has caused the disease. So in order to prevent you from having it again, which cells belong to it? Well, B and T lymphocytes. For sure, you know that the functions of B and T lymphocytes are different, right? So for example, what is the function of B lymphocyte? What does it do?
Differentiates into plasma cells.
Perfect, but say better. Yes, that's exactly what it does.
Produce immunoglobulins.
But that's the plasma cell who does it, right? So plasma cell makes antibodies, not necessarily the B. So the answer is perfect. It has to become plasma cell. So what is the function of a B lymphocyte then? And this is the most confusing question for all students because they always ask you, what's the function of B lymphocyte? And you always say it becomes plasma cell. The function is very simple. The function of a B lymphocyte is surveillance. It's just monitoring. Okay, it's just watching for antigens. It's just looking for them. And once it's found the antigen that it was looking for, Then this B lymphocyte gets activated and turns to become plasma cell, and this plasma cell will make the antibodies against that specific antigen that it has encountered, right? So very important to distinguish B lymphocyte and plasma cell. They have a very important difference. One of them is activated, one of them is not. Plasma cell is activated B lymphocyte. What has activated it? A very specific antigen, like a keyhole, like a key and a lock situation happened, right? So if I'm a B lymphocyte, I have a receptor, the antigen have come in and perfectly fit into my receptor, which has activated me, and I turn to become plasma cell. But the naive or baby B lymphocyte, it does not necessarily have a function. The only function it has is just surveillance. It's just looking for its antigen. That's what it's doing. It's just looking for it. T lymphocytes, we will talk about it today. We have different types of them. We have cytotoxic, we have T regulatory, and we have a T helper. But nevertheless, they, in common, what they have is that they kill things. Okay, so they go and they kill things. So how can I kind of divide, right, my two cells? Because obviously they do not have the same function. Again, B has to become plasma, plasma will make antibodies. T will go and kill things. So this is why your adaptive immunity can be divided into two subgroups. Okay, so there are two additional subgroups of the adaptive immunity. We have said that B lymphocytes, they get activated by its antigen. They turn to become plasma cell. This plasma cell will make antibodies. This has a name. It's called humoral response. So everything that I have just said, this is called humoral response. This is when, again, your B lymphocyte gets activated by a certain antigen, turns to become plasma cell, and that plasma cell makes lots of antibodies against that specific antigen that has it activated. So all of it is called humoral response. Everything that T cells do, and they do lots of things, is called cellular mediated immune response. Cellular mediated immune response. I haven't wrote here mediated, but it should be there. Okay, so everything in relation to T lymphocytes is cellular mediated immune response. Okay, cells, come back to this slide after you're done with this lecture. It's like an overview of what is happening. Okay, so simple overview of what is where, who does what, and etc. Now, this is going to be your second slide that is super important. So the first one was antigen. The second is going to be this, cluster of differentiation. So again, what is antigen? Could somebody tell me? So now that you're second year medical student, you say it as a second year medical student. What is antigen?
Anything called
Hmm?
a foreign marker.
Very good. This is a foreign molecule, a foreign marker of some sort, right? That can be on the surface of pretty much everything. Be that, I don't know, bacteria, virus, fungi, parasite, peanut, does not matter. Table, right? All of those things, they have markers on their surface, and it is considered to be antigen. But you remember we have said that my own cells, the cells in my body, they also have a marker, right? So they also tell me, what am I? Like, am I a lymphocyte? Am I a red blood cell or something like this, right? So the lymphocytes specifically and hematopoietic cells, their marker has a name, and it is called cluster of differentiation. Okay, so whatever marker is located on lymphocytes, whatever marker is located on hematopoietic cells, you call it cluster of differentiation. So for example, you know, we have lots of lymphocytes. We have B lymphocyte, cytotoxic T, helper T, Treg, right? Each and one of them will have a very specific CD on the top of it. And we represent that CD with numbers. So for example, if I am a B lymphocyte, on my surface, you will find cluster of differentiation 19 and 20. Because I see CD 19 and 20, I know that this cell is a B lymphocyte specifically. So what is the importance of cluster of differentiation? It is a very important cell marker. And where is it located specifically? Specifically on all types of lymphocytes and hematopoietic cells. Can somebody refresh my memory about hematopoietic cells? What are those? This is a separate topic in your embryology in history. It's the first 5 topics, hematopoiesis, in your embryo. Even though we learned it in histo last semester, but it will be asked in embryo this year. So for example, there's something which is called erythropoiesis, right? Thrombopoiesis, I don't know, granulocytopoiesis, monocytopoiesis.
Are they precursor cells?
Perfect, those are the precursor cells. And this is very important. So for example, what do I start with? I start with multipotential myeloid stem cell. Do you remember this? And this multipotential myeloid stem cell will give me 4 types of cells. What are those 4 types of cells? This is, as I have just told you, your red blood cell, your platelet, and your granulocytes, and your monocytes, right? Why lymphocytes are not here? Well, because they don't come from the multipotential myeloid stem cell. They have their own stem cell, which is called multipotential lymphoid stem cell, right? How do you call a process of making red blood cells? You call it erythropoiesis. And there's a bunch of steps here. For example, multipotential myeloid will become proerythroblast, which will become basophilic erythroblast, which will become normoblast, or another name orthochromatophilic, right? Another name polychromatophilic, then reticulocyte, and then red blood cell. Each of this stage will have a very specific CD. So if you want to be a hematologist, a doctor who deals with blood, you will know each and one of this CD. Each cell which is located along the pathway will have their own CD. So as Nasir has said, the precursor cell, right? So the cell that will give me my red blood cells or platelets or blah, blah, blah. They will all have on their surface some kind of marker. And that marker is called cluster of differentiation. If anatomy department likes you and they actually want to give you a point in the self-control test, you will get a very easy question. What does CD stand for? And what does CD stand for? It stands for cluster of differentiation. That's the answer. So it's 1 free point out of 10 questions, which is amazing. If you think it's very easy, it is, because in the lectures, they simply write CD. So it's all about you. Were you lazy enough to Google or not? What is CD? If you were lazy enough, then you wouldn't get the point what is CD. If you weren't, then you will get the point. So I hope that is clear that CD is kind of like a marker on very specific cells only. Which kind of cells? This is your lymphocytes and hematopoietic cells. So let's talk about lymphocytes in more details. For example, T lymphocytes. T lymphocytes are called T lymphocytes because they originate in thymus. Okay, so behind your sternum, there is an organ which is called thymus, and this is the place where you make lots and lots of T lymphocytes. If you're older than 25 years old, which I don't think any of you are, okay, so if you're older than 25, then you don't have thymus. I'm 28, I definitely don't have it. I have just fat behind my sternum where thymus should be. So thymus is very active when you're a child until you're about puberty or 18 years old. Starting from 18 years old until you're 25, it slowly starts to being replaced with fat. So more and more adipocytes start to appear within your thymus. And once you reach 25, until you die, slowly those adipocytes start to disappear. So this is why in your cadaver, when you open up the chest area behind sternum, you will find nothing. Why? Because all of your cadavers are old people. So if you were lucky enough to have a young person as a cadaver in your anatomy room, then you might find it. But majority of the cadavers are older people, so you will not see any thymus there. It forever have disintegrated into the thin air, because even those adipocytes which were replacing the thymus, even they have already disappeared. But you still have it, so it's good. So thymus behind your sternum actively makes T lymphocytes. T lymphocytes, as you remember, participate in the cell-mediated immunity, and they account for approximately 60 to 80 of the total amount of lymphocytes. So majority of your lymphocytes are T lymphocytes. We have three types of T lymphocytes. We have T helper, we have T cytotoxic, and we have T regulatory, or another name, suppressor. Do they have the same job? Absolutely not, right? So all three different types, they do different things. Yet on the surface of all three of them, on the surface of Treg and T cytotoxic and T helper, you'll find those CDs, CD2, 3, 5, 7. You need to know this. So this is why we call CD2, 3, 5, 7 as general CDs. They cannot be used to differentiate T lymphocytes. Why they cannot be used to differentiate? Because they, I repeat, they're located on all of them. It doesn't matter if it is T helper or T cytotoxic or T reg. On the surface of all of the three of them, you will find CD2, 3, 5, 7. Is it clear? So what can I use to differentiate? How do I know I'm looking at T helper or T cytotoxic or T reg? Additional CDs are used. For example, CD4 is only located on T helper, only. CD8 is only located on cytotoxic, and T reg only possesses CD25. So can somebody tell me all CDs, all of them, which are located on T helper?
CD 2, 3, 4, 5, 7.
And?
4.
And 4. And CD4. Okay, so I repeat that CD4 is the one that will tell you you're looking at T helper, because 2, 3, 5, 7 will not, right? It's general. Which CDs are present on cytotoxic?
2, 3, 5, 7,
2, 3, 5, 7, 8. And which CDs are present on Treg? 2, 3, 5, 7, and you add V25. So I repeat, CD3 can be used as a marker for differentiating different T lymphocytes, correct?
No.
Absolutely not, because CD3 is present on all three of them. Okay, it will be present on all three of them. So in another way, CD2, 3, 5, 7, I repeat, are general CDs. They're present on all T lymphocytes. It really does not matter. If anatomy department does not like you and they want to make your life hell, in the self-control test, you will see something like this. CD4 positive cell does this and that. What are they talking about?
T
Helper,
lymphocytes.
T helper. They're talking about T helper. CD8 positive cell does this and that. They're talking about cytotoxic. Okay, so don't forget. We have two types of T helper cells. We have T helper 1 and we have T helper 2. There are two different types of them. Somebody has a question?
Yes.
Yeah.
When you said CD4, CD8 positive, what does that positive stand for?
It means that you have it.
Okay.
I have CD4 and I don't have CD8 or CD25. If I say I'm CD25 positive, I have 25 and I don't have the others. I don't have 4 or 8. So if I'm a CD4 positive cell, I'm a T helper cell, because the only cell that has CD4 is T helper. So we have different T helpers, T helper 1 and T helper 2. In the immunology next year, they will give you additional CD, which will differentiate the two. For now, we don't care. So 2 types of T helpers, T helper 1, T helper 2, both of them still have CD4 plus 2, 3, 5, 7 as general ones, right? It is important to remember who do they interact with. Because you have to remember, T helper is this cell that the name tells you it helps, right? So how does it help? It calls for different cells. This is like a, I don't know, like a taxi control person. Like when you call 555, this is the cell that you call, okay? And you'll be like, can I have a taxi this way, right? So, and that taxi person will call another taxi so it can come and pick you up at a very certain address. So T helper is this, I think the word is dispatcher, like somebody who is like dispatching different taxis in the different directions, right? So T helper is the same. It's this one control center that just regulates who goes where and who does what, okay? Still, T helper 1 and T helper 2 will interact with different cells. For example, T helper 2 only interacts with B lymphocytes. So if for some reason you need B lymphocytes to come at a certain place, which T helper will be the one to call for B lymphocyte? It will be T helper 2. Okay, so T helper 2 will call for B lymphocyte. T helper 1 will call for cytotoxic, natural killer, and macrophages. Highly tested, both oral and self-control test, okay? So both of the T helpers, as you can see, they interact with different cells. So it all depends on who are you actually calling. So if you need macrophages, you won't call T helper 2, right? Because T helper 2 has no idea who is this macrophage you speak of, right? Instead, you need to call the T helper 1 and ask it politely to call for a macrophage. You can see here different cytokines. They are not important this year. They will be important next year. So for example, interferon gamma is a cytokine which is needed for macrophage. So how exactly does T helper 1 call for macrophage? It will secrete interferon gamma. And interferon gamma is like drugs for macrophages. Once they sense it, they try to come to the area where there's lots of this interferon gamma. Okay, I repeat, this year, you don't need to know the cytokines which are needed to call different cells. You just need to know who interacts with what. So please remember for the end of this class, okay, who does T helper 1 call?
Macrophages, cytotoxic T cells.
And natural killer cell. Who does T helper 2 calls?
Only B lymphocytes.
B
B
lymphocytes.
lymphocyte. And it is actually vice versa. If B lymphocyte needs help, right? If it needs help, it needs to, I don't know, call for some other B lymphocytes. It will go to T helper 2, right? So it won't go to T helper 1 to ask for it to bring more B lymphocytes. It will go for T helper 2. The same for macrophages. If macrophage has caught something, it will go to T helper 1 and ask T helper 1 to call for more macrophages to come. So it's vice versa. It's not only one-way interaction. Both cells can interact with one another. Cytotoxic T cells. It's in the name. Those cells are toxic to cells, right? So those are killers. Who do they kill? Virus-infected cells, cancer cells, or anybody who is infected with a microorganism, parasites, or even transplanted cells, right? So if, for example, I need a kidney, you guys give me kidney, my immune system will still attack that kidney because it's not mine, right? So just because I put it in my body doesn't mean that it will function properly forever until I die. No, it's still not part of my own body. So this is why I will need to kill it. Who will kill it? cytotoxic T lymphocytes. So don't forget that they are CD8 positive. Okay, so CD8 positive. And now we come for Treg, or suppressor T lymphocyte. Which CD it has? It has CD25, right? So what will be the job of Treg? Guess. A wild guess. What does regulatory suppressor T lymphocyte do?
Regulates the functions of the other T lymphocytes.
Perfect. So this is the cell who has one job, and that one job is to suppress immune system. This is the only cell which is capable of suppressing entire immune system. Why? I like to think of immune system as a very on edge kind of system. It's very nervous, right? Anything can set it off like this. Talking about like sometimes, I don't know, I eat honey or peanut and my immune system overreacts and thinks it's completely something else, right? And I have like an allergic reaction, right? So it's also where your immune system completely overreacts. Or I don't know, I simply have some bacteria. And yes, it's a bit difficult to kill that bacteria. Yet my immune system, I don't know, increases the fever so much to tell everybody in my body that we're under attack, that there's bacteria someplace, okay? So my body needs to know, so we need to regulate our nutrients to make sure that immune system has some for it, right? So as you can see, immune system is crazy like that. Like whenever it has one job, it has to protect you, and it will do anything to do just that. So even if it means killing you. So that's one way to look at it. So if your immune system cannot protect you, what is the solution? It's to simply ask you to die, okay? So hence the fever that rises uncontrollably, you need pills to control the fever, okay? So it's crazy, but it is like that. So this is why we need a cell that can definitely be like, take a chill pill, right? So just relax that yes, this is bacteria, yes, it's very dangerous, but you got it, you're under control. So suppressor T lymphocyte, as soon as your immune response begins, as soon as you encounter some kind of pathogen of some sort, Treg is there to regulate, to make sure that you don't pass a certain threshold, okay? So you don't overreact towards something. So who do you think, and pardon my French, who do you think fucks up in the autoimmune disorder?
Treg.
Treg. Treg in the autoimmune disorder does this. If I do not see it, it's not happening. Some cytotoxic T cell decided that kidney looks too familiar, it looks exactly as a certain virus it remembered. So cytotoxic T lymphocyte will start to overreact, it will start to attack your own kidney cells. Typically, T suppressor will be like, excuse me, what are you doing, right? So that's not how it's supposed to be. But in the autoimmune disorder, T suppressor simply looks away. It does not see that this is actually happening, okay? So this is why in the autoimmune disorder, it's not so much as who does the damage, it's rather who is not doing the one job it's meant to do, which is just regulating this entire process. In the book, they wrote you this part. I apologize, every year I forget to remove it. It's not needed, okay? So they never ask you. There's a specific marker, but it doesn't matter. It doesn't matter. B lymphocytes, why do we call it B lymphocytes? Because they originate in the bursa of birds. That's where they originally found them. Bursa is bone marrow. But then they realized that our own bone marrow also has B lymphocytes, okay? So it also has them. So the reason we call it B lymphocytes, because that's the place they are born. If T lymphocytes are born in the thymus, B lymphocytes are born in the bone marrow. What is their job? Surveillance, monitoring, until they meet their antigen, right? The one that perfectly fits onto their receptor. That antigen will activate them and they will become plasma cells and they will make antibodies or immunoglobulins against that specific antigen. 20 to 30% of them, okay? So if majority of the lymphocytes are T lymphocytes, 20 to 30 are B lymphocytes. Which CD markers they have? 9, 19, and 20. For self-control test, CD20. So they will say CD20 positive cell. CD20 positive cell is a B lymphocyte. Okay, so it will be B lymphocyte. Will it have 2357?
No.
No, because who has 2357?
T lymphocytes.
T lymphocytes. So don't forget that. CD2357 are only present on T lymphocytes, but not on B lymphocyte. So if they ask you this question, CD2 can be used to differentiate between B and T lymphocyte.
Yes.
Yes, because again, 2357 are present on T lymphocytes. CD2 is not present on B lymphocytes. So technically, I could use it to differentiate between T and B lymphocyte. Yes, there's nothing wrong with that sentence. This part in the oral can be asked. Highly doubt can be asked in the self-control test. It's quite difficult to ask this in the self-control test, but in the oral they can. So we have to understand the receptor of a B lymphocyte, and not just B lymphocyte. We have to see the receptor of a naive or inactivated, right, resting B lymphocyte. And we have to take a look at the receptor of activated B lymphocyte. Like, what is the difference in those receptors? So immunoglobulin, right, antibody. Whenever I tell you antibody, agree with me, you're always thinking that it is free, right? It's just like, I don't know, it's just within your blood, just flowing there. It's free. It's not bound to anything, right? But some antibodies are bound, and they are bound to B lymphocytes specifically. Okay, so some antibodies are not free, they are bound to B lymphocytes. And because they're bound to B lymphocytes, they act as receptors. So we have some antibodies which literally act as a certain receptor. One of those antibodies is immunoglobulin M, IgM. So imagine the following situation. I am a B lymphocyte. Okay, I am a B lymphocyte. I am inactive. I am naive. I am resting. I haven't met my antigen yet. On my surface, I have a receptor that tells me just that I'm resting, that I'm inactive. I haven't met my antigen. What does that receptor looks like? It looks like IgM. And what do you need to know about IgM is that it is a pentamer. Okay, it is a pentamer. It literally looks like a hand, pentamer. It has 5 things. So I repeat, if I am a B lymphocyte and on my surface you see IgM coming out like this, you see IgM coming out like this, what does that tell you? Am I active or am I inactive?
Inactive.
I am inactive. Have I met my antigen yet?
No.
No, I'm a naive B lymphocyte. My receptor is IgM. Take a look, it is like a hand. It's a pentamer. So what can I use this hand for? To grab something. And what can I grab with that hand? A certain antigen. It will come in and perfectly fit into my hand just like that. Antigen comes in and binds to my IgM. That's it, I caught my antigen. So what should happen to me next? I should become activated. So I change the way I am. So my receptor also changes. It's no longer a pentamer because I caught my antigen with it. Instead, it becomes a dimer. And how do we call an antibody which is a dimer? We call it immunoglobulin D. Okay, so this is immunoglobulin D. So I'm a B lymphocyte and my receptor is no longer IgM. It's no longer IgM. It's now IgD. And what does that tell you? If you see on me IgD, what happened?
Activate.
I'm activated, so I caught my antigen. What is the next step? Who should I become now?
Plasma
Plasma cell.
I should become a plasma cell. I should become a plasma cell. So let's repeat it. We have two antibodies. We have IgM and we have IgD. Which one is a pentamer?
IgM.
IgM.
Which one is a dimer?
IgD.
IgD. Both of them act as a receptor. Is it clear? So both of them are markers. Both of them act as a receptor. Which one is a receptor of a naive B lymphocyte? The one that hasn't encountered the antigen yet.
IgM.
IgM. So because it is a pentamer, it's very useful, right, to just grab my antigen like that. Because it is a pentamer, I grab it and that's it. I grabbed it. What should my IgM change to?
A dimer.
IgD.
So it becomes IgD. It changes to a dimer, it becomes IgD. So if you see IgD on the surface, the B lymphocyte is no longer naive. It becomes, as we say, mature. It's now active. It's now ready to become the one thing it should, which is a plasma cell, right? So to make antibodies. So I hope that is clear. Feel free in the oral to do this, honestly. They will love that, honestly. So like if you're do it, it just shows that you didn't memorize, that you actually understood what is happening. So I show a receptor and you tell me if I am active B lymphocyte or if I am inactive. Yes?
Active.
Active B lymphocyte. I'm active.
Inactive.
Inactive.
Okay.
Active.
Inactive.
Inactive. This is which antibody?
IgD.
IgD.
IgD. This one?
IgM.
IgM. Okay. If I'm a naive B lymphocyte, which receptor is my antibody?
IgM.
IgM.
M, because it's naive. I'm still naive. I'm trying to grab whatever I can. IgD, if I'm mature, I already caught it, right? So peace, I already caught it. We
I have a
talked.
question.
Yeah.
What happens to the IgD after the B cell becomes a plasma cell?
Perfect. You will change it to the type of antibody which is meant to kill that antigen. And typically it either stays as IgD or it changes as IgG. But you will learn it in immunology, okay? In third year, you will understand like what happens after that. For now, that's more than enough, but for you, you will either stay as IgD, so whatever your antibodies are coming out from you now, they're in the shape of IgD. Or you change to IgG, and then again, all of your antibodies coming out are IgG, okay? So the G one. So IgD or IgD, but typically they don't ask, so don't worry. But good question.
Really, the IgM and the IgD are only found on the B lymphocytes, like not any other cells?
Correct.
Okay.
Yes. No other cell will have them, only IgM and IgD, yes. Well, IgM can be on a macrophage. It can be. Okay, so in immuno, third year, again, you will learn that not only the B lymphocyte has it, also the macrophage. But for now, whenever you think of IgM or IgD as a second year medical student, you're still thinking about B lymphocyte. And then they will add to your knowledge that also macrophage also actually has IgM as well. Yeah. Okay, because think about it, IgM is very useful because it is a pentamer. It's very useful to grab antigen with it because it is a pentamer, because it's literally like a hand that is just trying to grab whatever it can. So whatever can grab antigen, such as your B lymphocyte or macrophage, it will probably have IgM on that surface. Natural killer cell. Least available cell, 5 to 10%. Okay, so the most is definitely your T helper, and natural killer is the least available. It's a little bit of both. It's a little bit B, a little bit T lymphocyte. It's a little bit of both. In which way? It also can kill things like a cytotoxic T lymphocyte. Okay, so it's also capable of killing things like cytotoxic T lymphocyte, but it doesn't arise from thymus. it doesn't, okay? So it's not the place where it comes from. Instead, it comes from the same place as B lymphocytes. It comes from bone marrow. So hence the similarity with a B lymphocyte, right? That they have a common origin with B lymphocyte, yet it can kill like a cytotoxic T lymphocyte, okay? So you see it's like a little bit of both. Who does it kill? Anybody, well, anything, any cell which is infected with a virus or tumor. Natural killer cell is a super efficient cell against cancer. It's so efficient, I'll tell you right now. So it's the one that does it amazingly. So whenever you think of cancer, you should be thinking about natural killer cells because that's the one that will definitely get rid of it. And the way it kills, I really love that as well because it literally shoots bullets, literally. Like it shoots little tiny vesicles at a cell which is infected, and those vesicles contain perforins and granzymes. And what do? They literally make holes in the plasma membrane of infected cell. If there is a hole, it means that cytoplasm can leak out, ions leak out, so that forces your cell to undergo apoptosis. So this is why natural killer cell, I repeat, it literally shoots bullets. Which CD it has, the one which is important for you, 56 or 94. They keep changing every year, okay? So we saw both. We saw 56 and 94 in the previous years. Okay, this is the third important slide of this lecture. Okay, so third most important slide of this lecture. We're talking about antigen presenting cells. I ask you to fully turn on your attention because I cannot repeat it twice. It's such a long story, so I will keep stopping to see that you get it, but I cannot repeat it twice. So in the recording it will be, but try to get all of your attention right now. So first of all, we're talking about something called antigen presenting cells. But before we start, can you tell me which cell has CD8 on their surface?
Cytotoxic T cells.
Cytotoxic, okay. Which one has CD4?
Helper.
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 Helper. So the way I remember, if it helps, CD8 is in cytotoxic. There's so many T, right? So many T. So CD8 is present in the cytotoxic. CD4 must be in the helper. But the way I remember, what does CD helper do? It asks for help. Help has how many letters? It has 4 letters. So this is why CD4, okay? So CD4 is helper. So, but the way usually to help you immediately is CD8 is cytotoxic. That means that CD4 must be T helper, it must be, right? We're introducing a new molecule, which is called MHC. MHC stands for Major Histocompatibility Complex. Whenever you're transplanting organs, this is what should be matched, okay? So MHC should be matched. So we should have at least 6 out of 8 compatibility, because in Immuno next year you will learn all about it. For now, it's just slow introduction. MHC. It's also some kind of marker, the same as CD, the same as antigen, right? It's also a marker. And we have 2 types of MHCs. We have MHC1 and we have MHC2. Who has MHC1? Any nucleated cell. It doesn't matter which kind of cell I have in my body, as long as it has a nucleus, on its surface you will find MHC1. So my epithelial cells, they have nuclei on their surface, MHC1. My, cardiac myocytes, again, they have a nucleus, MHC1. Skeletal muscle cells, nucleus, nuclei, MHC1, right? So you see that it definitely will be there. What about MHC2? MHC2 is only present on antigen-presenting cells. There's actually a question that they ask you in the oral. In order to be considered antigen-presenting cell, what should you have? you should have MHC2 on your surface. So the only way for you to be considered to belong to the groups of antigen-presenting cells is if you have MHC2. So I repeat, who has CD8?
MC1.
Cytotoxic.
Cytotoxic.
Who has CD4?
Helper.
T helper. Who has MHC1?
Nucleated cells.
Every nucleated cell, does not matter the place, right? As long as it's in my body. Who has MHC2?
Antigen presenting.
From now on, I call them APCs. Okay, so throughout our course, second ACT, third ACT, I say APC. APC means antigen presenting cell. Which cells are APCs? Here they are, highly tested. Macrophages, B lymphocytes, and dendritic cells. Okay, so those are the three APCs that you have in your body. I repeat, why is macrophage an APC? What did I tell you in the oral? They ask you, in order to be considered APC, what should you have?
MHC2.
MHC2. So I repeat, why is macrophage an APC?
Has MC2.
Because it has
MHC2.
Because it has MHC2. Why is B lymphocyte an APC?
Because it has MHC2.
Perfect. Why is dendritic cell an APC? Because it has MHC2. Is it clear? So all of them are antigen presenting cells because they have MHC2. Until this point, is it clear?
Yes.
Yulia, I just have a question.
Yeah.
When you say antigen here, you don't like, do we mean the self-antigen because these are cells of our own body? They're not like foreign substances.
I mean foreign, because don't forget that you're second year, right? So whenever you see the word antigen, you don't think about your own cells. You think foreign, right? So you think foreign. So let me play you a little something, okay? So let's imagine, how does it actually work,
How
right?
are like macrophages and B cells like foreign cells? They're our own body cells.
So let me show you what actually happens, right? So what does it mean, antigen presenting cell? What does it mean? So let's imagine the following situation. All of you are T helper cells. Each and one of you is a T helper cell. What do you have on your surface?
MHC1.
Well, MHC1, yes, because you have a nucleus probably, but what do you have on your surface if you're T helper?
CD4.
CD4. So each and one of you, because you're a T helper cell, on your surface you have CD4. Can we imagine that your right hand is a CD4? Okay, you're sitting right there in your own rooms, your right hand is a CD4. I am a macrophage. Okay, I'm a macrophage. Because I'm a macrophage, what do I have on my surface? I have MHC2, which is also my hands. MHC2 are also my hands. I'm a macrophage. So what is my job? I'm antigen presenting cell. Don't forget, macrophage is a phagocytotic cell. So I am perfectly fine just monitoring connective tissue. And then I see that there's lots of bacteria in one area. So what do? I chase that bacteria. Have you ever seen macrophages run after something that they're supposed to engulf? It's so hilarious because they're literally running. They're literally chasing something that they're supposed to engulf. Finally, I caught it. My phone is bacteria. Okay, my phone is bacteria. I'm a macrophage. So what do? I try to engulf it. I have engulfed it. Now my phone or bacteria is inside of me because I am a macrophage. I have fully phagocytosed this bacteria. As I have phagocytosed this bacteria, I realize that, listen, there's lots of them. I have engulfed just one. I don't think I can do this by my own. I don't think I can engulf all of them. So what do? I take a little piece, just the antigen of this bacteria, and I put it into my hand. What is my hand?
MHC2.
Perfect. It's MHC2. So right now the antigen, the receptor or marker of whatever I have engulfed, which in our scenario is bacteria, is located on my surface. It's on my hands. It's in my MHC2, right?
Mm-hmm.
What is my next step? I go to you. I go to my T helper, and I literally do this. Please, Miss Sir, me lady, look at this. Can you please look at it? Because you need to look at it. And you're looking at whatever I'm presenting to you. But what do you use to test it, to like take a sample of it? What do you use?
The bacteria on the...
Yes, so what do you use? Don't forget your T helper. What do you use to use my antigen, to test it, to sample? What do you use?
CD4.
CD4. You use CD4. You reach out with your hand, because you're a T helper, you reach out with your hand and you sense whatever I'm trying to present to you. So CD4 grabs whatever MHC2 has. Once you have sensed this antigen, you decide, yes, this is something dangerous. I should call for more macrophages so they can come and help you. Or you know what? What have you brought me? You can go back and do it yourself. Okay, go back and phagocytose it yourself. So what am I trying to tell you? MHC2. Who has MHC2?
Macrophages, B cells, and dendritic cells.
They are antigen presenting cells. Do you understand what this means? It means you show the antigen to somebody. To who do you show the antigen to?
To other lymphocytes.
To which other lymphocytes specifically?
T helper.
To the T helper. To the T helper specifically. This T helper cell is a cocky, lazy bastard who will not go by itself. It needs to be presented. It needs to be shown what is happening at the periphery. It's a king that is just sitting. It's a boss of the mafia that just does this. Go kill this, go kill that. I'm just sitting here like this. Who are the Santa's little helpers that actually do everything? It's the APCs, the antigen presenting cells. They go sample whatever is happening, put it on their little silver platter, which is the MHC2, and then go and try to show it to the T helper that please take it, tell me if we need to call for more cells or not.
So Yulia, they become antigen presenting cells once they're in contact with a bacteria or with like a foreign substance, and they take it and put it?
They're always antigen presenting cells because they have MHC2. As long as I make a macrophage, I always put MHC2 on their surface. So this is why we call those cells professional APCs. This is what they do. Take a look here, we have a B lymphocyte. Remember I asked you, what is the function of B lymphocyte? And we said surveillance.
Surveillance.
That's the second job, antigen presenting cell. B lymphocyte is also antigen presenting cell. That's what it do. As it's surveillance, it looks for anything it can present. It looks for the antigen that it can go to the CD4 and show it to. And be like, listen, there's lots of it. We might need some help with it. So can we do it again? And I do not want to hear Niyati or Nasir or Kavya. I want to hear other people and you tell me what is happening. So for example, let's talk with, I don't know, Simra. Your T helper cell, what do you have on your surface?
CD4 positive.
Yes, perfect. You're CD4 positive. I don't know. For example, Anya, you're a macrophage. What do you have on your surface?
APC.
You are an APC, but
Yeah,
what
you
do
are
you
an
have
APC.
on your surface?
MHC2.
Perfect. You're a macrophage, so you test what is outside. You saw lots of bacteria. You engulf it, right? You phagocytose it. Muhammad, what do you put into the MHC2?
The bacteria?
Not the entire bacteria, just a piece of it. How do we call that piece? Don't forget, which kind of cell are you?
Helper.
Well, you are, we used to be, but we're still talking about macrophage. Macrophage is which kind of cell?
Antigen presenting cell.
Presenting cell. So what do you put into the MHC2? What are you presenting?
Antigen.
Antigen. So antigen is this little tiny piece of whatever you have engulfed, be that bacteria, virus, or whatever. Okay, Ryota, who do you go with those Who do you go with this MHC2 that has the antigen? Who do you go?
To helper T cells.
Perfect. And what will T helper use to sample this antigen?
To recognize MHC2.
I'm a macrophage. My hand is MHC2. My phone is antigen.
CD4
You
plus.
are T helper. So what would you use to take my phone?
CD4 plus.
Perfect. You will use CD4. Okay, so you will use CD4. So CD4 will be used to sense this. So forever, thank you very much. Forever in your brain, forever, whenever you hear MHC2, immediately what should pop into your brain is CD4. They're always interacting. There's a literal handshake that is happening between macrophage and a T helper, between B lymphocyte and a T helper. And if those cells are using MHC2, right, to shake, the T helper uses CD4 to shake. We call them antigen presenting cells. A better way to say is that they are professional antigen presenting cells. Why? Because that's what they do. That's what macrophage, B lymphocyte, and dendritic cell do. They go around, sample things, right? Phagocytose them, put little pieces of those things on their MHC2, and just keep running to T helper and be like, is it dangerous? Can I have some help? What do you think? Can you help? Blah, blah, blah. That's all they do. That's how they earn their bucks, by constantly going to the T helper and asking for help. But we also have non-professional antigen presenting cells. Okay, we also have non-professional. And which cells are non-professional antigen presenting cells? It's any nucleated cell. Any cell that has a nucleus can be considered as a non-professional APC. Why? Because it simply has MHC1, right? So don't forget, who has MHC1? Every nucleated cell, right? So every nucleated cell has MHC1. So let's imagine the following situation. I am an epithelial cell. I was infected with influenza virus. Okay, I'm infected. I'm in the esophagus. I have been infected with influenza virus. I need to put a marker, right? I need to say that I'm infected. So what do? I put a little piece of my influenza virus onto my MHC1, right? Because I'm epithelial cell, I have a nucleus. If I have a nucleus, I have MHC1 on that surface, on the surface of me. And what do I use this MHC1? As a platter on which I can put whatever has infected me. Who will come and sample it?
Cytotoxic T cell.
Very good. Cytotoxic will come and sample it. And what will it use to sample this antigen?
CD8.
CD8. So again, MHC1 only interacts with CD8. And do you see what is the difference here? CD4 will not go. It won't go to sample. It will sit back like this and wait until APCs will come to it and show it whatever is infecting things, right? Cytotoxic is not like that. It's a hard worker. So as it is monitoring, it is just going at it. If it sees that some cells on their MHC1 have, I don't know, an antigen of some sort, that they have bacteria or virus, this cytotoxic T cell will come, sample this antigen and be like, you know what, yeah, you're definitely infected, so how about you just die already, right? So apoptosis, let's induce the apoptosis. Phew. So a question for you. I hope that was clear. So now let's test your knowledge. Macrophages can only present antigen to the T helper cell. Correct or incorrect?
Correct.
Macrophages can only present antigen to the T helper. Why is it incorrect? Do macrophages have a nucleus?
Yes.
Yes. Do they have MHC1 on their surface?
No.
Why
Yes.
not? They have a nucleus. And MHC1 is present on any nucleated cell. Any, it doesn't matter. So I repeat, do macrophages have MHC1 on their surface?
Yes.
Yes. Why do they have MHC1 on their surface? I repeat. Because
They're nucleated.
they're nucleated. Because MHC1 is present, who can macrophage also present antigen to?
Cytotoxic.
To cytotoxic. So it is incorrect to think that antigen presenting cells can only present antigen to the T helper. Mostly, yes. Mostly they present to the T helper, but they can still, and they're still capable because they have a nucleus to present antigen to the cytotoxic T lymphocyte. Is it clear? Do red blood cells have MHC1?
No.
No.
Okay. Do platelets have MHC1?
No.
Yes. Why do the platelets have MHC1? Think. Whenever you describe a platelet, what is a platelet? Is it a separate cell?
It used to be a big one.
It is a fragment of a bigger cell. Of which cell?
Megakaryocyte.
Perfect. Megakaryocyte, is it nucleated or not?
Yes.
Yes. So why do platelets have MHC1? Because they're simply a fragment of a bigger nucleated cell. So this is why platelets do have MHC1. That's the question I was asked by Professor Rosa, actually. Do platelets have MHC1? But luckily I remembered that they're just fragments. So I was like, yeah. And she was like, whoa, you just blew my mind because not everybody knows this. But yeah, okay, so again, red blood cells, it's a bit different because there we actively squeeze the nucleus out, right? Every cell loses its nucleus as it tries to enter into the sinusoid of the bone marrow. For platelets, it's a bit different. It's just one cell, megakaryocyte, it just bursts, right? Undergoes, as they say, fragmentation. So platelets do have MHC1. So I hope that was clear. So take a look at it again as you do the recording. We talked about this. We talked about this. Okay, so now we talk about lymphatic vessels. Lymphatic vessels are very much similar to your blood vessels. They also have tunica intima, media, and adventitia. The only difference is that lymphatic vessels are way more permeable than blood vessels. They allow even proteins to get in or get out. Okay, so they're way more permeable. Another difference is that lymphatic vessels actually possess valves. What is the purpose for those valves? To establish unidirectional flow. So don't forget is that your lymph flows from the connective tissue towards the bigger veins, right? So right here we have the venous angle. I'm not sure if you have learned it in the anatomy yet, where the subclavian vein kind of goes at a 90-degree angle with the internal jugular veins, and this angle is the place where all of the lymph will eventually end up. So in order to make sure that all of your lymph will end up in veins, you introduce the valves, right? So they make sure that only unidirectional flow. Here we have a lymph node, just like a stop, like a filtering area, right? So like a lymph node. Two types of lymphatic vessels, afferent and efferent lymphatic vessels. So A are bringing in, right, the lymphocytes, and E are taking them out. So this is very important. So afferent lymphatic vessels is the entrance point and the efferent is the exit. Only about 20% of lymphocytes are using this route. So 20% of lymphocytes are actually using lymphatic vessels in order to transport around body, to come to the lymph node and etc. 80% will use a blood vessel to get to the lymph node. And we call this blood vessel, we saw them last semester, we call them HEV, high endothelial venule. Okay, so high endothelial venule, HEV. This is the name of the blood vessel that lymphocytes use in order to get to the lymph node or to other lymphatic tissue like spleen. What is high endothelial venule? It is a post-capillary venule, right? So it is a post-capillary venule. Remember we have like, I don't know, large elastic artery, a muscular artery, small artery, arterial, 3 types of capillaries, and then after capillary there is post-capillary venule, then there is muscular venule, then small vein, medium vein, and then the large vein, right? So that's the entire pathway around your body. So a very specific type of a post-capillary venule, which is called high endothelial venule, is located only in the lymph node and spleen. Okay, and we call it high endothelial venule because its endothelial cells are not squamous, but they're rather cuboidal. Okay, so normally endothelial cells should be squamous, but here they are cuboidal. Okay, so 8% use this. The way we call this process of lymphocytes returning to the lymph node, okay, so they're coming back to the lymph node. It's a very cute name. We call it homing, like you're coming home, homing. Okay, so homing, that's the name of the process when lymphocytes use the blood vessel to return to the lymph node and lymphatic vessels. So simply the act, that lymphocytes return to the lymph node, we call it homing. Here we're talking about antibodies, very nicely written for you here, everything. So I just want to kind of get your attention to the most important things. So first of all, there are 5 types of antibodies. Okay, so you already saw this one, IgM. Remember it? This is a pentamer, and what's up? Which kind of cells have it?
Inactivated B cells.
Very good. Inactivated, or we call it naive B lymphocytes, right? So the ones that haven't encountered their antigen yet. This is the biggest antibody there is. IgM is the biggest because it is a pentamer. It's like a hand ready to grab everything, wherever. IgD, remember?
Also B lymphocytes activated.
But activated, or we call it mature B lymphocyte, right? So the one which has already encountered its antigen. So this is the one which will change its receptor from IgM to IgD. IgA, you remember?
Present in all of our secretions.
Perfect. So it's all of your secretions. Doesn't matter which, here they are. IgG, this is the smallest antibody. Okay, this is the smallest antibody. It is so small that it easily passes placenta. So for example, you have an embryo, right? There is an organ called placenta, which kind of filters the blood, right? So of the embryo, brings the good things, takes away the bad things, and etc. So maternal IgG, because they're so small, they can easily cross placenta and get to the embryonic blood, and as a result, establish passive immunity. Your embryo doesn't have immune system. Its immune system consists of the maternal IgGs, which are actively protecting it from whatever pathogens can be there. However, this IgG gives passive immunity only until the first month of birth. So as an embryo, you have IgG, maternal IgG, because it crosses placenta, right? Very tiny, comes to embryo's blood, protects. And in the first month after you're born, you still have it. What is another antibody that will give passive immunity to baby now, not embryo, baby? This is going to be IgA. As long as you breastfeed the child, you will be given the IgA with the milk, and this IgA will be very important. This is actually one of the reasons why it is important to breastfeed the child. Formula has very little amount of IgA, so there is a higher chance that the baby can get sick, okay? So because of it, so it's very important to naturally breastfeed the child because of this. IgE, allergic reaction. So take a look, who does IgE bind to the mast cell and basophils, and it forces them to release their contents, which is the histamine and heparin. And hence you have this vasodilation, I don't know, some kind of itchiness, right? So, and etc. Okay, let's talk about thymus. Thymus is a primary lymphoid organ. The place where it is superior mediastinum. This is the upper third of a space which is located behind the sternum, right? So here we have our sternum. The space behind it is called mediastinum. There are three of them: superior, middle, and inferior mediastinum. Super active in children, right? So this is why it is very important to be sick as a child. To, I don't know, eat sand, drink dirty water, because your immune system will encounter more and more pathogens. It will make more T lymphocytes, which will protect you further in life, in the adult life. So this is why in the first world countries, like, I don't know, America, UK, Germany, the children are sicker than in the third world country, because in the third world country, the water is unfiltered. You eat, I don't know, dirty vegetables, dirty fruits, right? Sand, I don't know, something like this, the dust, all of this stuff. So your immune system is much stronger compared to the babies which were born in the first world country. So that's why it's actually a downside of living in America or UK or Germany, is that you don't have a very strong immune system. So if you go, I don't know, to third world country, like, I don't know, Africa or Russia, you'll get sick like this, okay? You'll get sick like this. So make sure that you know this. Okay, two lobes divided into many lobules. If you see the word lobules, that means that it has capsule. And from capsule, there are septa. Remember when we talked about salivary glands, right? So there's some lobules there. They are there because of the capsule and septa. Each of the lobule has an outer region, which is much darker. It's called cortex, and an inner region, which is much lighter, which is called medulla. Why is that? Cortex has more cells than medulla. Okay, so the same situation, embryological origin, third pharyngeal pouch. In embryo, we will talk about it. Okay, so just third pharyngeal pouch, brackets, endoderm. Okay, so endoderm. Your thymus has endodermal origin. Okay, only has efferent lymphatic vessels. Why? Why does thymus has only efferent? No afferent are there.
Because it produces the lymph. The lymph.
Perfect. So you need to remember the following. It is a primary lymphoid organ, thymus. It is a central lymphoid organ. This is the education area. This is the place where you're making T lymphocytes. So what's the purpose of afferent lymphatic vessels if there aren't any cells to travel in it, right? So this is why primary lymphoid organs never have afferent. They only have efferent. The developing T lymphocyte is called thymocyte. Okay, so this is how we call it. The baby T lymphocyte, which is present within your thymus, we call it thymocyte. Those thymocytes are surrounded by supporting cells, and those supporting cells are called epithelioreticular cells. So there are two types of cells in the thymus, the baby T lymphocytes, which we call thymocytes, and supporting cells, which are called epithelioreticular cells. Here I'm telling you the different stages of thymus, that there's thymus epithelialis during embryonic life, lymphaticus. This is the slide that we have, okay? So this is a baby's thymus, a piece of it, and has lots of lymphocytes, so we call it thymus lymphaticus. And adiposus, this is a different stage. It begins after puberty. It only starts after puberty. You start to see some adipocytes there. When is it completed? Closer to the age of 25. Okay, so that's when it fully turns to a fat pad. And from 25 until you die, even this fat pad disappears. It completely disintegrates. Okay, epithelioreticular cells. There are 6 types of them. Okay, we will talk about them right now. But first, it is epithelioreticular cell, right? So it's partially reticular cell. So what should this reticular cell do? It should make reticular fibers, which are the collagen type 3, which are dominant in reticular connective tissue, or another name for it, loose connective tissue, right? Collagen type 3 is a dominant fiber in loose connective tissue. Even though this cell, this supporting cell, is called epithelioreticular cell, reticular connective tissue is not there. Sorry, my voice. Reticular connective tissue is not there. Okay, so you will not find it there. I repeat, it's called epithelial reticular cell. It should be making reticular connective tissue. It should be making collagen type 3. It should be making loose connective tissue. You won't find it there. Loose connective tissue is not present within the thymus. So 6 types. Easier to describe them on the slide. Here we have our thymus. This is a capsule, cortex, medulla. Okay, so cortex is darker, medulla is lighter. Why? Because cortex has more cells, medulla has less cells. Type 1 epithelial reticular cell is at the border between capsule and cortex. It's right here. So that's where the type 1 epithelial reticular cell should be. What is its job? Its job is to have some kind of junction. It should have this occluding junction. If junction is there, then it is a barrier. It makes sure that none of the things can get in, because within the capsule we have blood vessels, and that could be problematic because with blood vessels, pathogens can come and they can diffuse this area and they can make your baby T lymphocytes bad children. They will introduce to them viruses and will say, listen, this virus, you shouldn't attack it. You should actually avoid it. Okay, so that could happen. So in order to avoid that, you put a barrier. This is the type 1 epithelial reticular cell. It forms a barrier and it prevents the unwanted exposure of the baby T lymphocytes that haven't learned what is good or bad yet. Type 2 and type 3 epithelial reticular cells are within the cortex. They're in the cortex, okay? So type 2 and type 3 epithelial reticular cells are within the cortex. Take a look. They have MHC2. They have MHC2. Who had MHC2?
Antigen presenting cells.
Very good, such as?
Macrophages, B cells, dendritic cells.
And who do they present antigen to?
T helper.
They're going to be presenting to the T helper cell. So what do you think about type 2, 3 epithelial reticular cell? What will they use this MHC2? Will they use it to present antigen to T helper? No, because this is thymus. There aren't any T helper yet, right? So there isn't any cells there yet. There's just baby T lymphocytes. So who are you presenting this and for what reason? The reason that type 2 and 3 have MHC2 on their surface is for education. They're going to put the antigens onto this MHC2, and they're going to shove those antigens to the face of the T lymphocytes for one reason only, and that reason is for education, to show them what is antigen. Okay, so the T lymphocytes can learn what is that. We'll talk about it in a sec. So you can think of type 2 and 3 epithelial reticular cells as your teachers, as your professors, or as me, okay? So it's somebody who is teaching you something. Thymic medulla will possess the following epithelial reticular cells. So right at the border between cortex and medulla, right here, we'll have type 4 epithelial reticular cell. The job will be the same, to make a barrier, to make sure that none of the pathogens can get in. Type 5 is just within the medulla. Job is to compartmentalize lymphocytes, to kind of form groups, to make sure that all of those cells are there. And finally, type 6, this is the one that they typically ask. Type 6 epithelial reticular cell is very special because it possesses keratin. Can you tell me who else has keratin?
Epithelial cells.
Very good.
Have
Who else?
keratin.
Who else?
Dead cells.
Epithelial cells that are dead cells, sure. Who else has keratin?
Every cell has keratin.
Not necessarily.
Those
Okay.
which don't have nuclei.
Yeah, the dead cells, they definitely have lots and lots of keratin. Yeah, but the keratin that we speak is a polymer. It's like lots of it. So which cells have lots of keratin? Why do I take like shampoo with keratin? Or I don't know, nail polish with keratin? For which reason? So I can support my hair and nails, because my hair and nails are also dead cells and they also have lots and lots of keratin. You have never ever known that hair have keratin? For sure you have known this. I don't believe it, that you have never heard that nails and hair have keratin, that they're filled with this like intermediate filament called keratin, that they have lots and lots of it. So now you're going to add the fourth thing, which is the type 6 epithelial reticular cell. For some reason, for some unknown reason, nobody knows why, the cell will also have keratin, will have lots and lots of keratin. And the aggregation of type 6 epithelial reticular cells, which have keratin, we call it Hassel's corpuscle. So Hassel's corpuscle is this, I don't know, little tiny circular structure which possesses type 6 epithelial reticular cells which have keratin. Here the book talks about what could be the reason for having keratin, because I mean thymus is not your hair, it's not your nail, it's not your skin, so why should it have this hard keratin? Just, I don't know, it's just a guess, you don't need to know this. Blood-thymus barrier, a very important slide, because don't forget we're trying to protect our baby T lymphocytes, right, from pathogens. Where do pathogens come from? They come from blood. So what are the layers of the blood-thymus barrier? The first one is called endothelial, right, so endothelial cells of a blood vessel. The second is the basement membrane on which endothelial cells are sitting. The third are pericytes. Do you remember pericytes? What is their job? From last semester, what is the job of pericytes? What do they do? Why do they have them around blood vessels?
To give nutrition?
I can get nutrition from blood directly. If I'm endothelial cell, just facing the blood, I can get it from there. So they are contractile cells, because you have to remember that those cells, pericytes, are only present in the capillaries and post-capillary venules. What is the same thing for both of those vessels? What unites capillaries and post-capillary venules? What do they have in common that other vessels maybe do or they do not? What's the purpose of pericyte? We have said that it's contractile cell. So in other blood vessels, what is used to change the diameter of a blood vessel? What could be used to contract?
The walls of the blood vessel itself, like the
Yeah,
thickness.
so what is in that wall that can be used to contract?
Parasites.
Smooth muscle.
That's pericytes are only in capillaries or postcapillary venues. We're talking about arteries, I don't know, arterioles. What is there that is contracting and as a result changing the diameter of the wall?
Smooth muscle.
Smooth muscle.
Smooth muscle, in which layer is it?
Media.
Tunica media. So I repeat the question. What unites capillaries and postcapillary venues?
They don't have smooth muscle.
They don't have tunica media. It's not there. They don't have smooth muscle cells. So what is the purpose of pericyte? The purpose is the same as the purpose of tunica media, to contract. So if I don't have tunica media, I need to have some kind of cell which will do just that. And this is the pericyte. If it dilates, the blood vessel dilates. Sorry, if the pericyte relaxes, the blood vessel dilates, right? If it contracts, the blood vessel, I don't know, squeezes, right? So it changes its diameter. Very special about pericyte. Which kind of cell is it? We talked about it last semester as well. So it's a very special cell. What is so special about it? Yes, it contracts. Yes, it's present in those blood vessels, but what is so special about it?
Fibroblast type.
Is a remnant of a mesenchymal cell. So it is a what? Which kind of cell is it? It is a remnant of mesenchymal cell. Which kind of cells are mesenchymal cells?
You mean the ectomesenchymal cells?
No, I mean actual mesenchymal cells, the ones which are in mesenchyme. What is mesenchyme?
Connective tissue
Not just connective
of
tissue,
the embryo. Of the embryo.
embryonic connective tissue, and possesses very special mesenchymal cells. Which cells are they? Which kind of cells are they?
Fibroblasts.
No, they are, but they have very special property. They're pluripotential stem cells. So pericyte is a pluripotential stem cell. Okay, so it is a pluripotential stem cell. So all of you have failed. All of you would have repeated this year, $17,000 each and one of you. So it is a pluripotential stem cell. So it means that it can become anything, anything. There's some, I don't know, experiments where they take out pericytes and they literally grow cartilage out of it, a cell that simply, I don't know, contracts to dilate, or the opposite of a capillary, right? So I don't know, to squeeze it out, it can be used to grow cartilage. It's insane because it is a remnant of a mesenchyme, because it is a pluripotential stem cell. Outside of the pericytes, we have perivascular connective tissue, which possesses macrophages. Those macrophages are the most important part of the blood-thymus barrier, right? So they are the ones which will be actively phagocytosing any pathogen which is trying to get into your Dieldrop T lymphocytes. Next layer is the type 1 epithelial reticular cells. So don't forget, they also have junctions, the same as your endothelial cells. They also have a barrier, right? So a tight junction. So very difficult to squeeze it out. And last year, they have added the 6th layer. Apparently, type 1 epithelial reticular cell is sitting on a basement membrane. So there is a basement membrane of the type 1 epithelial reticular cell, which cannot be visible on, like, I don't know, any kind of drawings, but it is there. Okay, so it is there. So this is a blood-thymus barrier. You need to know it's the thickest barrier of all in your body. Nothing is more than 6 layers. Even your brain is 2 layers. Blood-brain barrier is just 2 layers. Blood-thymus barrier is 6 layers, okay? Blood-air barrier is 4 layers. Blood-placenta barrier is 3 layers. So this is the one that is like the thickest. And now we finally reach the 4th slide that is very important, right? So the first one, as you remember, is the antigen. Second is the CD, cluster of differentiation. Third is APC, and now the T cell education. I highly doubt, like, it's very difficult to make self-control test out of this question, but it's not impossible. And anatomy department is known to do the impossible things. So this is why you can see a question that can be taken from here, from this part, from how do you educate the T cell. But again, it's very difficult to ask it. You're gonna see what do I mean. So we start with the T cell education. Basically, how can I make my T cells? Don't forget we have 3 types. We have T helper, T cytotoxic, and T reg. In second year, you begin with the knowledge that T-reg are already formed. We have already made them. The first T cells to make are the T regulatory cells. How do you make them? We don't talk about it in anatomy department. We talk about them in immunology. There is a whole process of how do you first make the Tregs. But you're starting T cell education with a thought that T regulatory cells are already present. They're already there, okay? So let's talk about how can I make T helper or T cytotoxic cell. I have 2 cells, T lymphocyte number 1, T lymphocyte number 1. It is number 2, it is undifferentiated T lymphocytes. It's not a cytotoxic, it's not a T helper. So what is my first stage to do to those baby T lymphocytes? I push them through a double negative stage. Double negative stage. Double negative stage is, the name tells you, they lack something, right? So let's not focus on what do they not have. Let's focus on what they do have. So what do you gain during double negative stage as a developing T lymphocyte, you gain general CDs. Can you tell me what are the general CDs?
2, 3, 5, 7.
Very good. So I know I wrote here 2 and 7, 3 and 5 as well. So this is a developing T lymphocyte on its surface during double negative stage. You will see CD2, 3, 5, and 7. The same is here, CD2, 3, 5, and 7. Why do we call it double negative stage? Because the two CDs, which are important, such as CD4 and CD8, were not present, were not there. So that's why you call it double negative, right? The second stage is double positive stage. So what is happening here? You do get CD4 and CD8, but the key word is not or, it's and. So all of your T lymphocytes express both. They express both CD4 and CD8. Again, CD4 and CD8. So hypothetically, both of those cells are both T helper and T cytotoxic, right? Because they have both of the CDs, CD4 and CD8. So why do we call it double positive stage? Because both of the CDs are expressed, I repeat, CD4 and CD8. I like to think about it as, I don't know, us in high school. It's very rare when you know what you want to do in high school. Like you're aware fully that you're going to be a doctor or a dentist, or I don't know, something like this. Like if I remember myself, I wanted to be everything. I wanted to be an ambassador, a teacher, a veterinarian, a doctor, a blogger. I wanted to be everything. So the same are your T lymphocytes here. They want to be everything. They want to be both cytotoxic and T helper. Why to decide now, right? So I would rather just express both of them, CD4 and CD8. So when you reach double positive stage, can you tell me which CDs you have on your surface? All T lymphocytes, which CDs they have on their surface?
2, 3, 5, 7, 4, 8.
Very good. CD3, 5, 7, right? So 2, 3, 5, 7, 8. So 2, 3, 5, 7, 8, 4. So all of them. So all general and all of the specific. The next stage is positive selection. Okay, this is the next stage. It is done by type 2 and 3 epithelial reticular cells. And try to understand, like try to tune in right now. So what happens? You are my baby T lymphocytes, all of you. I am type 2 or 3 epithelial reticular cell. I'm your teacher. My hands are MHC2 molecules, okay? And what do? I take an antigen that was given to us through evolution, that we know this bacteria causes this disease in this particular area where you're from. I don't know, like for example, E. coli in Siberia. Literally in our gene, we already have antigens of E. coli because of how often we get it. So I take this antigen of E. coli, which is a foreign bacteria, put it into my MHC2 and shove it into your face as my baby T lymphocytes. I show it to you and I say, this is foreign antigen. Do you get it? And all of my baby T lymphocytes like, yes, we got it. And then I take antigen of our heart or our brain or our, I don't know, kidney. And I again shove it to your face and say, this is self antigen. Do you get it? And all of you are supposed to like, yeah, we got it. If you got it, you passed positive selection. In positive selection, the baby T lymphocytes are being introduced by type 2 and 3 epithelial reticular cells through the MHC2 molecules. They're being introduced the foreign antigen and the self antigen. And only those cells which recognize both are allowed to pass. They pass the positive selection. So let's imagine, I don't know, again, type 2 epithelial reticular cell. MHC2 is right here. I put foreign antigen. I shove to my baby T lymphocyte Niati. And I say, do you understand that this is self antigen? And Niati is like, yeah. And then I put another foreign to my MHC2, and I go, and this is foreign, it's not ours. Do you get it? And Yati is like, I don't understand. I understand this is self, but I don't understand this is foreign. So what happened? Apoptosis. You don't get it? You're too dumb to be a T lymphocyte, right? So you're going to die. So only, I repeat, those are allowed to pass that recognize both the antigen, which is on the surface of the cells in their body, which they're located, and the foreign. Where does positive selection happen? In the cortex, right? So in the cortex of the thymus, you will find positive selection. Once the cells that have passed positive selections leave the cortex, they enter medulla, they begin another stage, which is called negative selection. And what happens in the negative selection? Negative selection, you do not have there type 2 or 3 epithelial reticular cells, because this is medulla, right? It's an already different area. This is medulla. Which cells are here? Here you have Treg, T regulatory, and they do the same thing. What do? They introduce the two antigens to you, again using MHC2 molecules. Okay, so they're using MHC2, the Tregs, they show again the foreign antigen and the self antigen to the baby T lymphocytes. But now this happens in medulla. Why do we call it negative selection? Because the T lymphocytes are supposed to forget the self antigens. So how does it happen? Treg shows foreign antigen to the Treg, to the T lymphocytes. And T lymphocytes are like, yeah, we definitely saw it in the cortex. I recognize that this is foreign antigen. And then it again shows it the self antigen. And the T lymphocytes, which are successful, which are allowed to pass the negative selection, they do this. They're like, this is the first time I'm seeing this. I have never seen this self antigen before in my life, even though I just passed it, right? I've never seen it before in my life. If you do this, you are successful, you pass the negative selection. Can you tell me, why is it important for the baby T lymphocytes to forget the self antigen?
So that they don't attack it when they become mature. And
Perfect. So we call it autoimmune disease, right? So if my T lymphocytes recognize the self-antigen, they will attack it. You guys must understand, if I recognize the antigen, it's not good, okay? It's the same as police sees a criminal, they're like, yeah, it's not good, I must catch it, I must do something. If you recognize it, it's not good. So if your T lymphocytes recognize self-antigen, the markers of their own body where they're located, they would want to attack it, obviously. So this is why it's going to be autoimmune disease. So do you see who regulates this process? Treg. So in the autoimmune disorder, what happens? The problem is from day one, from the moment of embryo. Okay, so from the moment of embryo, when you're making T lymphocytes, Treg looks at the baby T lymphocytes, shows them the self antigen, and the baby T lymphocyte react to that self antigen. They're like, yeah, that's a self antigen. And the Treg is like, well, good for you, off you go, because you know it. So this is your autoimmune disease. Instead of saying like, wait, you aren't supposed to recognize it, right? They're like, amazing, you passed, off you go. Okay, so that's the major. The last stage, single positive stage. So what happens? Randomly, you get to decide. Do you want to keep CD4 or CD8? This process happens randomly. Randomly, your baby T lymphocytes are asked to drop one of the CDs, okay? And they choose which one they want to drop. If I drop CD8, I have CD4, so I'm T helper. If I drop CD4, I have CD8, so I'm cytotoxic. So this is the overview. I hope it was clear. Watch it in the video again. Just one more thing here. How many cells in percentage do you think pass? All of it. They reach the single positive stage. How many cells in percentage pass?
2%.
1.
Amazing. 2%, 2%. Okay. Typically students tell me like 40. I'm like very optimistic. 20%, very optimistic. It's 2%. Only 2% of the T lymphocytes pass through this entire stage. This is literally Sparta. Whenever I don't want to study for step 1 or step 2 or step 3 USMLE, I'm like, no, how can I do this? I'm not smart. I don't think I can do this. I always remember T lymphocytes. If they pass through this, I can do anything. Literally, there's nothing that I cannot do, okay? Because if 2%, the only 2% success rate, can you imagine? It's insane. If they can do this, I can do, or you can do anything. So which cell is super, super busy here? Macrophage. It's busy swiping the yard because so many dead cells are there. It's insane. So macrophages are busy. Okay, let's quickly do the slides. Let me just quickly tell my other group that I'm late. Is the 2. Okay, so here's your thymus. This is thymus lymphaticus H and E. Okay, so this is going to be thymus lymphaticus H and E. So what do we have? Here we have capsule. Here's your thymus. The darker areas are cortex. The lighter areas are the medulla. In your histopracticals, they say that this is pseudolobulus, that the thymus is pseudolobulated, and they're right. Because in order to be considered as a true lobule, the outer area has to be cortex and the inner has to be medulla. We have some lobules which are only cortex, only. So this is not a true lobule. So it is a pseudo-lobulated appearance because there's definitely no appearance like this, that outer is the cortex, inner is the medulla. So here we are. This is your capsule. I repeat, this is the darker area is the cortex and the lighter area is the medulla. You can see within medulla this kind of looking structure. What is this?
Hassel's corpuscle.
Very good. This is the Hassel's corpuscle, right? So this is the type 6 epithelial reticular cells, which are filled with keratin. Don't forget, it has no function, right? So this area has no function whatsoever. Now, this area, the thicker, this is called trabecular, and the thinner is called septa. Okay, so this is just histological differences, specifically in the thymus. The area which is much thicker, this is what we call trabecula. The area which is much thinner, this is called septa. So again, darker area.
Cortex.
Guys, come on. Cortex. What happens here? What did I just say? What happens in the cortex? I just said it. Weren't listening. Positive selection. What happens here?
Negative selection.
Negative selection. So this is the medulla, okay? And here we have the negative selection. This is the Hassel's corpuscle up close, right? So this pinkish area, this is going to be keratin. This is the hard keratin, which we typically see in skin, nails, and hair. Okay, 2 types of cells you see. Here, the darker cells, those are developing T lymphocytes, right? So those are the thymocytes. Every cell which has a darker nucleus, this is the developing T lymphocyte or thymocyte. The cell with a lighter nucleus, this is going to be epithelial reticular cell. Okay, luckily we don't have to identify the type, which type is it, but judging from location, because this is medulla, it's within the medulla, this could be type 5. Okay, so this could be type 5. So again, this
Trabecular.
trabecular, very good. This
Septa.
septa, very good. This
Cortex.
cortex with positive selection. This
Medulla.
Medulla.
medulla with negative selection. Which cell regulates positive selection? Which cells are responsible for it, basically?
Epithelial reticular. Type 1 and 2.
Which one?
Type 2 and 3.
Type 2 and 3. Type 2 and 3, because they have MHC2 molecule, right? Which one regulates the negative selection?
Tregs.
Treg. This is Hassel's corpuscle with keratin, right? Here, the darker cells, I repeat, thymocytes, developing T lymphocytes. This cell with a lighter nucleus is the epithelial reticular cell, and this cell with this kind of structure, this is a macrophage. It has a halo, okay? So this is how you can kind of recognize it. This whitish area, this is the halo. And if the halo is present, that's it, this is a macrophage. So this is a macrophage right here. Okay, so let's quickly do this. Lymphatic tissue, diffuse lymphatic tissue. So here we are, this is mucosa. Okay, so this area right here, this is mucosa. And you know, mucosa consists of the epithelium plus underlining lamina propria. So whenever they say diffuse lymphatic tissue, what you mean is lamina propria. So this entire area right here. Another word for it is mouth. Another name for it is mouth. So the same way you can describe it, you could say it is mucosa, associated lymphatic tissue. As you can see, it is diffuse, right? All of the lymphocytes are everywhere within the connective tissue. But we have some areas where lymphocytes kind of aggregate, where they concentrate. It's also part of the mouth, right? And we call the structure lymphatic nodule. Do not confuse lymphatic nodule with a lymph node. They are not the same thing. Lymph node is a separate structure. It is encapsulated. It is a separate thing on its own. Lymphatic nodule is simply concentration or aggregation of lymphocytes within mucosa. And you remember that mucosa is simply a layer of a certain organ. It's not even an organ by itself. It's just a layer, the inner layer of a certain organ. So nodule, I repeat, is not encapsulated. It doesn't have a capsule because it is simply concentration of the lymphocytes within the mucosa. Mouth is too general. It's a general name. It depends on the organ you're in. This area, for example, is in the GI. Right? It is in your large intestine. So we call it not mouth, but gut, because it is gut-associated lymphatic tissue. So what is gut? Gut is mouth in the GI. That's what gut is. For example, mouth in the bronchus will be called bult. Bronchus, associated lymphatic tissue. Mouth in the skin is called salt. In the nose, it's called note. So you get it, right? So a common name is mouth, but depending on the area where you're at, you change the name. Here we're talking about lymphatic nodules, okay? So the areas of concentration, right? So just an area where lymphocytes concentrate. I would rather describe them on the slide, okay? So on the slide themselves. Large intestine, H&E. Here we have mucosa. You have said mucosa consists of epithelium plus lamina propria. So what is another name for all of it? What is another name that can be used for all of it if we're talking about immune?
Diffuse lymphatic tissue.
Very good. Another name?
Mouth.
Mouth.
But don't forget you're in the GI.
Gut.
Gut will be more specific. They will literally ask you. General name is definitely mouth, okay? So general name is definitely mouth, but specific name for it is going to be gut, okay? So this is the specific name. So what is this?
Epithelium.
Very good. And what is this?
Lamina propria.
Another name?
Gout.
Gout, because we're in the GI. Another name? Diffuse lymphatic tissue. And here you can see a bunch of lymphocytes together. How do we call it?
Lymphatic nodule.
Very good. We call it lymphatic nodule. Older professors, like Professor Clara or Professor Anterl, will not call it nodule. They could say follicle. This is the lymphatic follicle. Okay, so do not get confused if you hear or see two words. The same thing. This is a lymphatic nodule. Now, this type of lymphatic nodule is primary. So this particular one is primary. One. Why? Why? Because it is uniform. Okay, it's throughout looking the same. Here we have B and T lymphocytes. They're looking the same. There's the same. Take a look at the next slide. Here is your nodule. Even with this magnification, you can see that there is something in the center, right? Something pale in the center. How do you call this area? This area is called germinal center. Okay, so this area is called germinal center. How do you call the area which is outside? This is known as the mantle zone. This is the mantle zone. Is it primary nodule? Well, definitely not, because it has this germinal center. We call it secondary nodule, secondary follicle. So can you tell me quickly, what's the major difference between primary and secondary nodule?
Non-uniform and uniform.
Again?
Primary is uniform.
Say better, too general. What does secondary have that primary does not?
Secondary has
Germinal center. That's the word that you should use. Secondary nodule has a germinal center in its center. Okay, so it has the germinal center. It has this pale area. What are here? Here we have B lymphocyte. So germinal center is B lymphocyte dominant. This is where the B lymphocytes are. Mantle zone, T lymphocyte dominant. So let's imagine what actually happens here. I'm a B lymphocyte somewhere in my lamina propria. Okay, so chilling, nothing is happening. And then I notice that there's lots of bacteria in my epithelium, right? So there are lots of them. So what do? I take a sample of it, because don't forget that I am a B lymphocyte, right? So I'm an antigen presenting cell. I have MHC2 molecule, right? So what do? I take a sample of this bacteria, and I notice it's very, very pathogenic. So what do I need to do? I need to find the closest nodule there is, the closest. And typically this nodule is primary, right? It's very primitive, it's primary. So I repeat, here I am, B lymphocyte, caught something very suspicious like bacteria. So what is this B lymphocyte doing? It's returning to the primitive, it's returning to the primary nodule. Who is this B lymphocyte trying to show the antigen to?
T helper cells.
To the T helper. Within this primitive nodule, primary nodule, there's some T helpers sitting here. And this B lymphocyte shows the antigen to it. And T helper is like, yeah, this is definitely very, very pathogenic. So I give you blessing to proliferate. Okay, off you go, proliferate. Make as many copies as you want of yourself. So where would you do those copies of yourself? Where do you proliferate? Why go far? Why can't I do in this nodule specifically? So you start to see that B lymphocytes are actively proliferating. They're making lots and lots of replicas of themselves here. How do you call the area where they're actively replicating?
Germinal center.
Germinal center. Okay, so germinal center is very important. If you see it, it means infection. If I see it, it means that's where my B lymphocytes are proliferating. Something has caught their eye. Okay, so they need to proliferate. All the T helpers are going to be outside. They're going to be in the mantle layer. Is it clear? Okay, so I'm going to stop the sharing. I'm going to stop the recording.